Well, thank you everybody for joining us for our latest LABEST PACE event. I'm Mark Sosnowski, Senior Director of Biopharmaceuticals at UCLA TDG. And I'd especially like to thank our local institutional collaborators of USC, City of Hope, and Caltech who are joining with UCLA today for this event. And essentially with the goal of is really to provide an overview and of career options for the biopharma postdocs and grad students who are deciding on career options. And today's focus will be on search and evaluation. Essentially, how do the biopharma folks scout, evaluate, and select research innovations in order to drive collaborations for new therapies for patients? So today's panel, I'd like to introduce Susan Begelman, who's the Vice President of U.S. Respiratory Franchise at Genentech. David Crosby, Academic Research Collaboration Lead at Bristol Myers Squibb. Jessica Droge, Vice President, External R&D Amgen. And Barbara Sosnowski, Vice President and Global Head, External R&D Innovation at Pfizer. And she also is a Board of Directors member for UCLA TDG. So the way our format is laid out for this program today is each of our panelists will have 5 minutes to present an overview of their career, how they got into search and eval, attributes for success, followed by a few panel questions to drive discussion. But then we will use the second half of this program for breakout rooms to really drive engagement and allow our postdocs and grad students to interact directly with our panelists to understand opportunities in this field and, to decide if this is something of interest to them. So to kick it off, I'd like to ask Barbara if you would start with your introduction of your role and background in this career.
Great. Yeah, well, thank you for the invitation. It's a pleasure to be here. I try to go back to my high school, which was an all-girls school, to talk a little bit about science and the passion that I have for the science. So it's always rewarding to kind of give back. So happy to be here. Just to let you know, I have a PhD in genetics from Johns Hopkins University. It was on C. elegans. And then I moved from there to do my postdoctoral fellowship at the Salk Institute, also doing genetics in Drosophila. And I realized when I was in both my graduate and postdoctoral area that I really wanted to do translational biology. I wanted to focus on getting drugs into humans. And so I then applied in biotech in San Diego. I was in the San Diego area at the time. And I spent an enormous amount of time trying to convince people that Drosophila and C. elegans were relevant to human genetics, which was a kind of a challenge. But anyway, got into biotech, and had been in biotech for many years in various different positions. So I got to see a lot of scouting efforts. Also, I was VP of R&D for a biotech company at one point. I was chief operating officer. I was also CEO of a new company that I started at some point, and then I sold that company and went to that company to run their clinical program. So I varied from being a bench scientist all the way up to being the CEO and running the show and raising money, capital, things like that. So it was a lot of fun, and then I was doing search and evaluation all the time, as well as doing the internal R&D work that was necessary, writing grants, things like that. And Pfizer called about 11 years ago, and I thought, why not? I've taken risks before. I thought this would be a great opportunity to lead a group of individuals that is solely based on external evaluation. And so I run a group of 18 people that scout across the world, across all 5 therapeutic areas that Pfizer has, and also the large molecule, small molecule, so all our partner lines that fit into the therapeutic area research units. And we are global. Majority of us are in the US, but we have people in Australia, China, and in Europe. So it's a great group of people. It's exciting because I get to see new science every single day. I'm challenged by it, which I love, but it's a real passion to be able to see new opportunities, work with academics, and use their their great new science that they have, and then translate that into a work that can be done at Pfizer. So, in a nutshell, that's my background.
Great, thank you. Susan, would you like to provide your background?
Sure, thanks so much for having me. I actually bring a different background, perspective, career trajectory, and even look at sort of these acquisitions in my current career. So I too was always interested in the sciences, but more on the clinical side of the science sector, and ultimately got my MD from what was then Hahnemann University School of Medicine, it's now called Drexel College of Medicine, and followed the path of a clinical career, moving into residency, fellowship, in a long-standing clinical practice. I was at the Cleveland Clinic all in for 10 years. I'm a vascular medicine physician, and I was in the Department of Cardiovascular Medicine. And just through a connection of someone I trained with, first under and then was peers with, moved into the industry over 14 years ago now, and really picked up more on the late stage side of the trajectory of drug development in our industry, and worked in a small company in phase 2, phase 3 work before moving over to Genentech over 12 years ago now. And my career has actually moved along with the life cycle. So I worked in late stage clinical development across a variety of therapeutic areas, and spent a good amount of my time now in medical affairs, the area of the pharmaceutical industry, biotech, that's really focused on the post-marketing space. So also on the science side, doing clinical research and the like, until just about 6 months ago, where I moved over to the business, and I currently lead our US respiratory business. So we have several marketed, a couple marketed products, as well as one going into phase 3. And what's sort of interesting in terms of, I guess, this conversation is really a little bit about how we sort of view those types of acquisitions and drug development from the commercialization perspective. So along my career, particularly on the science side, I was involved in a few due diligences, and even now, members of my organization, I lead a large team of individuals across a variety of backgrounds, but also ultimately it's a cross-functional team with scientists, business, access individuals, is we get asked from a US affiliate perspective as part of a larger global organization, so Genentech's the US arm of Roche Pharmaceuticals, as to what these molecules, if they are successful, what's the business need, what's the impact on the clinical side, how would it fit into physician decision making, how does that look different from what's currently available today, does that bring value to patients and to providers. So it is a little bit of a different look-see, although I have a much broader remit than specifically sort of R&D and acquisitions. Thanks so much again.
Great. Dave, you to UCLA.
I was scared though.
Be curious to hear your background as how you got into your role with academic liaison.
Sure. Thanks so much, Mark, and thanks so much to the TGG group for hosting me here today. This is always a lot of fun.
What the webinar is about, but
Let's talk about yourself for a good hour straight. So about myself, I'm Dave Crosby. I live in San Francisco, California. In summary, I got my PhD at UCI, Go Eaters, in 2010. That was in retrovirology as an HIV guy. I started up my postdoc at UCSF in 2010 and finished that up in 2014. I had two foci there. I had my own postdoctoral research program around RNA metabolism and HIV. And the other piece I picked up is I booted up and operated a virology core lab as part of the UCSF and UC Berkeley P50 funded HARC Center, or the HIV Accessory and Regulatory Complexes Center. Boy, that's a mouthful. But long story short, I got a really good taste of what it meant to be part of a really broadly collaborative academic environment. And I just loved it. I really loved it. So in 2014, the medical science liaison role kind of came to me through a side door. It's kind of a funny story. Family member, my sister-in-law, introduced me just one night randomly over dinner. Said, you know, Dave, you ever think about becoming an MSL? My first question was, what's an MSL? I don't really want to work in sales. Thank you very much. And she continued to explain it to me what the role really was. around communicating, the basic and the clinical profile of the company's products, engaging deeply with all sorts of different external stakeholders in a specific therapeutic area. And the more MSLs I spoke with, the more I kind of fell in love with the role. I got my first MSL role with a itty bitty company, well, at least at the time, Pharmatherapeutics, based in New Haven, Connecticut. Sadly, the product that I was looking to launch failed a gonorrhea trial. Boy, that's a different story all in itself. But I was back on the market. And in 2015, I luckily found my way through my professional network to Bristol Myers Squibb, or BMS for short. I got my first role as a virology MSL, which kind of made sense with my background. So it was good to step foot back in virology. Although I didn't stay there long. In 2016, BMS's virology department kind of closed up shop. I stepped into rheumatology, where I supported a suite of projects, namely Orencia for rheumatoid arthritis and several other rheumatic diseases for a few years. I became a trainer. I sat on the leadership team for that group, developing training and development projects for the team at large nationwide. And then most recently, as July of this year, so I'm definitely the most junior in terms of experience with trying to evaluate collaborative opportunities with external stakeholders. I stepped into the role of academic research collaborations, ARC for short. So as an ARC, I have 3 goals. Number one, first and foremost, I try to pull through BMS's data generation strategies with 3 key academic institutions, namely, of course, my favorite, UCLA, UCSF, and Washington University in St. Louis. Now, this spans the full portfolio, right? So all the therapeutic areas that BMS has got a big toe in, and the full developmental pipeline. So everything from discovery through commercialization. So it's quite a large swath, but with relatively few external stakeholders, so to speak. I also try to collect and share insights about the institution's research expertise and capabilities, and also their perspectives on BMS's evolving portfolio of assets, which has evolved dramatically in the past couple of years with the acquisition of Celgene and now with the acquisition of Myocardia. So the company I started working with all of 5 years ago is very much a different company now than it was in 2015. And last, and also very important, is I do my level best to try to pull together the full BMS medical matrix team that touches these 3 key institutions. What am I trying to do? I'm trying to promote a cohesive face of BMS with this institution, with either its departmental leadership in a specific therapeutic area, with its operational leadership in various clinical trial units, with members of the C-suite chancellery and that sort of thing. And that's a lot of fun. It's a lot of building relationships, leveraging relationships. In many ways, it's very similar to the MSL role. But I just have a very different objective with what I'm trying to do now as an ARC as opposed to an MSL. And that's my career in a nutshell. Thanks so much for having me.
Yeah, thanks. And Jessica, please,
Hi.
about your background.
Great, and thank you for having me. It's fascinating to hear everybody else's backgrounds as well. I'm a huge fan of the UC system. I just have to say I'm a double UC graduate, went to UC San Diego for undergrad and UCLA for grad school. So I wish that I had participated in something like this as an undergrad or grad student back in the day. And I don't even know if it was available to me. I was not aware of it. So I think these types of forums are phenomenal, and I hope the grad students appreciate it, and hopefully there's something that comes out of it for them. But I'll say it would have changed potentially my career path. When I was an undergrad, I was a biochem and cell bio major, and I worked then after graduating in a lab. I wanted to be a neuroscientist and worked in a neuroscience lab, and realized very quickly that I didn't enjoy that type of bench science. I was in a basement, and there was internal fights about grants, and I was working with mice 24 hours a day, and it just wasn't my cup of tea. And so I thought, hey, this is not, I don't want to be a PhD in neuroscience. Even though I find the information fascinating, this is not the lifelong career that fits my personality. So I actually love data analysis, I love math, I love science, and I love statistics. And at the time I said, oh, okay, I'll do epidemiology, and I love infectious diseases. So I went to UCLA and got my PhD in epidemiology for infectious diseases. And while I was working there, I got a job with the Centers for Disease Control and they put me in charge, mistakenly or not, for one of their clinical studies. And this was a Southern California study that enrolled a large number of pediatric patients with Southern California Kaiser, testing an experimental measles vaccine. And so I, without any experience, was put in charge of managing this clinical study. I loved it. I loved working with the CDC. I loved learning all the drug development from a development perspective, the process. So after that study completed, and that was 4 years, that's what I did my dissertation on, I stayed with UCLA Center for Vaccine Research, doing clinical trials with experimental vaccines of all different types. Loved that. Then on one day, a recruiter called me and said, hey, you know, do you want to run the clinical affairs department at a cardiovascular startup? Sure, because I have no experience in that. That sounds wonderful. So moved down to San Clemente and worked with a small startup company for several years on a cardiac device. So device, not a drug, cardiovascular, not any areas, but you get these same skill sets that transport with you, you know, and you can adapt and you can learn. And if you love science and you love clinical development, you'll be able to pick that up. And then from that to several other different companies, and then eventually I landed at Amgen and did clinical studies at Amgen. I was in the clinical department for several years. And then I was, you know, wanting to grow a little bit more, and the business development group offered me a job in the same therapeutic areas that I had been supporting in clinical development. So it was an easy transition. I found the work fascinating because as opposed to drug development, if you're working on one drug or maybe a couple drugs, it's a very slow process and it's years. And in business development, when you're in search and evaluation, I head up now the group of search and evaluation. We cover all the therapeutic areas of Amgen, and it's from, you know, technologies all the way to marketed products, collaborations, et cetera. But you can't dive in deeply and stay for a long time in any one particular program, but you get to see the cutting edge of innovation around the world in every different area, meet wonderful people, learn so much. You're basically in grad school for the rest of your life, and it's a phenomenal career. And it is not, and I'm so glad to see, you know, that bench science PhDs, the MDs, and it's when I was a kid, I thought when you were a PhD, you had to stay by the bench. If you were an MD, you had to stay in the clinic, and that's not the case at all. But those type of degrees open doors for you to do so many different things. So that's who I am in a nutshell, and I'm happy, you know, during this program to answer any questions.
Well, thanks. Yeah, and thanks for those great overviews. I guess for the panel, the first question would be for our audience with search and eval, and you touched on this, Jessica. What are the attributes for success that you should have? Conversely, what if you don't have this, you know, run and hide, don't even consider this, or this is a burden within that career?
I'm happy to
Please,
take it, and others. You have to have a good personality. You have to be able to work with all sorts of different people. You have to have the patience of a saint, because you will be looking at different things, and the company that you work with may change their mind in terms of strategies, et cetera. You have to wait for the right time to engage, but you have to be curious. You have to be objective, and you really have to be strategic. How is this maybe very early on idea going to translate 10 years from now? What will the world look like 10 years from now? But those just are a few things.
Yeah, I would add that you need to be able to willing to shake people's hands and meet them. I know we can't do that now, but certainly when we were at meetings, you know, you can't be afraid to go up and introduce yourself, talk to them about the science. For my particular area, since I do, we had change in scopes, that I do everything from idea all the way up to candidate stage, which is where your IND enabling studies start to take place. So I'm very much engaged with the scientist early on, and so you have to be, even if you don't know it, yeah, you have to ask. Everyone wants to say something about their science. They just want to engage with you. So I think, not being, it's helpful to be extroverted, and being able to go that way.
Dave, anything further?
Yeah, I would say to echo other people's comments, knowing the difference between being subject matter competent and a subject matter expert. The vast majority of things I feel I know enough to be dangerous in most situations, hopefully, and hopefully the people I work with feel the same way. But honestly, if you have a strong desire to need to know all the facts before you enter into a conversation with somebody, this may not be the right role for you. Yeah.
I was going to interject on sort of a slightly related perspective is you're not always going to be right in the end. And so you're ultimately have to make a decision based on the best available information at the time. And I've worked on programs and acquisitions that have now come to us to either try and develop and commercialize, and they just fall apart, and it happens sometimes. And so I think as clinicians, as scientists, you have to be okay with imperfect information and use the best information that you have available at hand to make a decision. And you don't have endless amounts of time to do it. That's the other thing, is right. It's not like sort of seeing your experiments play out. If it takes 3 years, it takes 3 years. You really have a short period of time. And the other comment I would say, it's very much about teams and tapping into the subject matter experts that you have, really, and taking all that information together. And so when I've sat on some of these teams, in my experience, I love the fact that I come together with a very disparate group of people I ordinarily wouldn't work with at the company. And for a period of time, we're a team, and then we disband. And if I was asked to do something again, it's a whole different group of people. And so that's sort of a very interesting dynamic response when I know in my own career journey, early on in the sciences, you're used to working with sort of the same people in that same cohort consistently. And this is a bit of a different thing, which I find far more exciting in many respects.
Great, thanks. I think for the next question, again, to our audience, is what are the entry points for searching eval? What is the pathway? And I think related that too, with all four of you coming from really biopharma market leaders, how do each of your companies engage with this talent pool, in terms of fellowships, things of that nature that would be a next step of advice for them? And I guess I'll start with Barbara.
Yeah, I think to engage in this, when we go to scientific meetings, I think some of the areas that, as I was working and I find new ideas and this was exciting and I bring it back, you start out kind of putting your toes in the water. And then when you realize that door was wide open and you really like doing it, then you would actually engage kind of in a much larger fashion and, kind of get recognized for the fact that, you're out there scouting and you're going to be the eyes and ears for the company. So I think, you can start out slowly, and that's what I did. It was, and I think the way we also within Pfizer, we have sabbaticals, and people shadow us, and so that they get to learn, because we want to make sure that people at Pfizer don't feel pigeonholed into their space. So we offer those kinds of things for people to, maybe advance their career or change careers. We also offer internships for graduate students, as well as we have a big postdoctoral program as well for for if you want to be a postdoc within Pfizer. So there's a lot of different avenues, and we're actually trying to reach in earlier to go back to schools to help with the diversity and inclusion within Pfizer. So that's a huge effort that our CEO is very determined to make sure that we do right on that.
And I'll add also another way is, if you're a grad student and you enjoy what you do, I would suggest, doing some of that in the real world as a career initially too. It really helps when you're in business development to understand how the other functions work that you will lean on for their perspective in terms of evaluating a product. So if you come from that world, be it research or clinical or one of the other functions that's going to be, part of the drug development plan, you will understand the challenges that can occur maybe for a very early program, all of the pitfalls, all of the potential tests and assays that should be done. Not saying that they have to be done before a partnership is, but you'll really understand how much work and what really the feasibility is. On the other side, if you want to go on the clinical route, you partake, you maybe do involve yourself in some drug studies, you'll understand, how much work goes into designing those studies, how much importance the design has on the eventual label that drug may or may not get. So be in the real world for a while outside of business development. That would be my strong suggestion. That being said, as Barbara mentioned, Amgen has intern programs too for grad students and undergrads and a postdoc. Do that, absolutely, but don't then just stay in BD from day zero. I just find BD people are much better off if they've had some other type of career exposure.
Susan?
Yeah, I would say I certainly agree with what was said. And there are many ways to get to an end goal, right? So careers take all kinds of trajectories. I think philosophically, I always say if you can get some real world experience in some capacity, regardless of your role in pharma, it will make you a much better contributor to really understand that pathway. But one could even come in through the science side into a company like ours, and through that experience, learn about that whole drug development pathway and what it takes to commercialize a medicine, and then get into sort of the business development side of the business, either as an employee or get that experience and leverage that to understand. I certainly, having been a clinical practitioner making decisions for patients, having participated in clinical trials as a clinician at the institution, then designing and running trials, and now commercializing medicines, it is amazing how difficult it is to get from a great scientific concept to actually a medicine that treats patients or some sort of technology that advances care. And the more that you can better understand that, and so it doesn't have to be that you come in right through that avenue, you can come in through a piece of that process and work your way into the business development function, which a lot of people have done as well, as you've heard here.
Great. And Dave?
Yeah, I love the entrepreneurship route suggestion there. I did the Idea at IPO program at UCSF years ago as a postdoc and just loved it. To your point, Susan, there's a lot of ways an idea can die, and a lot of them have nothing to do with the science. And that could be, I'm sure UCLA, Mark, must offer a program that's right in terms of scope. I should probably know this. But it's a great way to just put your big toe in the fast-moving current that is trying to take an idea from off the page and actually, commercialize it. It's tremendous experience. The other thing I would say is just advocating from my own experience, look into the MSL, working in field medical. Number one, lets you test drive just how extroverted you are, how fearless you are on the phone, how willing and comfortable you are with being in communication situations where you don't know all the facts, where you don't know the therapeutic area that well, where your PhD basic science background may not translate intimately in conversations with a community rheumatologist. Boy, I've had a lot of experience with that. And just to gain an appreciation for what the patient provider experience really is. At the end of the day, you're developing products that your customers are potentially your family and your physician, and to develop a real appreciation for their concerns and what they want. And then I think from the MSL role, it gives you a good entree into a lot of the internal workings of the business. You don't necessarily get deeply involved, but you see a lot of the gears turn. And if you feel like you've got the soft skill set and just the appetite for going further, then you can do it. Plus, you can also work from anywhere for the most part as an MSL. There's territories all over the country, so you don't necessarily have to relocate at first. But down the line, in most circumstances, you find it.
Well, that's great. And thank you for all of your insights and guidance and advice. And now I'm going to turn it back to Marvi, because for the second half of our program, it's time to go into the breakout rooms.
Hi, everyone. Thanks again for joining us. So the breakout rooms, there are 4 panelists, so there will be 4 different breakout rooms. There will be a TDG team member who will be moderating within the breakout rooms. So I think we're ready to go. So I will push the button and you will automatically be going into your breakout rooms. We will not be convening back into the main room. We're just going to end it after the breakout room. So again, thank you so much for attending. We appreciate it, and we hope that you further your careers with all of the talks and all of the insights that you get today.
Yes,
Here we
thank
go.
you.